Welcome to Spatial Conversations, a webinar series by Millennium Science and 10x Genomics, where we feature some exciting new science and discoveries in multiomics, spatial multiomics space using 10x Genomics spatial platforms. I am Karthik Pillala from Millennium Science. I'm the account manager for 10x Genomics covering Queensland and WA. Today we have two speakers. The first will be Brett Kennedy. Brett is 10x Genomics Associate Director for Spatial Sales for APAC. He'll briefly introduce the newly launched spatial platform called Atera. Following this, we have our guest speaker, Stephanie Brauer. First, I welcome Brett to tell a little bit about Atera. Over to you, Brett.
Thanks, Kartik. Thanks everyone for joining today. Look, I'm quickly showing Xenium because obviously all this work is being run on our current Xenium platform. It's been a really exciting addition to the technology, and for some reason my slides aren't progressing. Thank you. And now it's being utilized by so many users to generate this exciting data, which we're hoping to share with you today. But I wanted to quickly talk about the next generation of technologies, which we've recently announced in April at AACR. It's not available yet. It's not shipping till towards the end of this year, but we're really excited by our new Atera platform. And what is Atera? Atera it really expands on what's possible with spatial biology, with a high sample throughput system, whole transcriptome gene expression levels, a really large imaging area with using off-the-shelf slides, with sensitivity and specificity comparable to single-cell RNA-seq, with the added flexibility of up to 3K-plex panels and a roadmap of new features. I mean, in terms of what that means, we've got some data sets online, which I encourage you to have a look at. But just quickly, this was a breast carcinoma sample where we generated around 23 individual clusters, which you can see spatially arranged on the slide at the right. What was really exciting about this sample, though, was that we were able to actually identify amplifications from expression of the genes within this region. When we plotted that back against this sample, that had spatial context to this amplification. And when we looked at the expression across those different cell types, you could really see it was clustered. The genes in this region of the amplification really clustered around high-grade DCIS cells. So a really interesting opportunity. We worked then with one of our customers, who actually generated the first Atera preprint article where they actually looked at this data, and they did laser microdissections of these regions and were able, using whole genome sequencing, to actually validate the presence of this amplification actually in the tissue. And they looked at immune profiling around different clones of this amplification. So a really exciting application, and the QR code is there for anyone interested in getting more information about that preprint. The other exciting thing about the preprint was they also looked at how Atera was able to identify rare cell types within this sample. We know that from our own work that we were able to identify dendritic cells in the tissue, which these are quite hard to identify. In the preprint, they actually looked at this and they were able to identify plasmacytoid dendritic cells, which is a really rare cell type, more commonly found in blood and lymph tissue. And they actually validated this using an Orion multiplex IF system. As we mentioned, Atera is whole transcriptome with the ability to add on up to 1K of target genes, as well as modular RNA panels. It has significantly higher throughput. You can do up to 800 1 centimeter squared whole transcriptome samples per year. It has higher sensitivity than the current Xenium 5K platform, about 2.5 to 3 times. And it comes in at a much lower cost. So we're really trying to push the boundaries to enable the technology. It is comparable in sensitivity to single-cell RNA-seq, with 3 orders of dynamic range and a low noise floor, so a low false discovery rate. And we've shown its sensitivity is comparable to Chromium Flex. You can get a wide range of cell type, tissue types all on the one slide. We have one processing protocol, and you do get this wide range of expression based on the cell type itself rather than the limitations of the technology. Because it is a new launch, we're really excited to show that there's also a long-term roadmap for the technology around more panels for whole transcriptome and other species, targeted application panels, software improvements. But the big three are we plan to add the ability to do RNA sequencing, targeted RNA sequencing with the current instrument technology. We're going to add workflow automation to speed up the detection, the preparation of samples and slides. And finally, we're going to add same cell protein profiling so you can add RNA and protein onto the same slide for the multiomics data, which everyone wants today. We are in the process of working with large numbers of early access users for the technology. This is another publicly available data set, for those interested, that came from the Allen Institute. Again, the QR code can take you to that. So I just want to finish to say that we're really excited that we think Atera will really help redefine biology with high plex, high sensitivity, and high throughput. And with that, I'd like to thank you and move to the talk.
Thank you very much, Brett. I'm very excited to introduce our speaker today, Stephanie. Stephanie is a PhD candidate in the Cancer Neuroscience Laboratory at QIMR Berghofer, supervised by Dr. Lachlan Harris. She completed her honors in Harris's lab, investigating calcium-binding protein in neural stem cell quiescence. She is now taking her work forward into her PhD, aiming to identify and characterize novel regulators of neural and glioma stem cell quiescence. So today she will be talking titled Lullabies of the Aging Brain, Niche Signals that Enshroud Neural Stem Cells. A very, very exciting title. And before the talk, I just want to tell you all that you can post questions to both our speakers using the Q&A button at the bottom, or you can unmute yourself and also ask the questions. This is Spatial Conversations. We're really excited to have some good conversations with you all. With that, please take it away, Stephanie. The floor is yours.
Thank you, Karthik. Can everyone see my screen and pointer?
Yep, I can.
Yes, okay, lovely. So as Karthik said, my name is Steph, and I'm excited to be able to share with you one aspect of my PhD project this morning, which does look at lullabies in the aging brain, the signals that enshroud our neural stem cells. So as a little bit of background for everyone, in the adult brain, neural stem cells really do facilitate our ability to remember and learn. They function in our sensory perception and they maintain our cognitive functioning. And they do this by generating new neurons that run those circuits in a process called adult neurogenesis. Now, the population of stem cells in the brain is incredibly finite, and these cells don't have the ability to renew indefinitely or replicate indefinitely. And so to last our entire lifetime without becoming replicatively exhausted or acquiring replication-induced DNA damage, stem cells will actually enter a reversible state of G0 cell cycle arrest, or quiescence, quite early in life. And that really does protect them. So we have a pool of neurogenic cells in our brain over the whole lifetime. Now, it's really important that stem cells enter this quiescent state, but quiescence can kind of cut the other way and become a bit of a problem because as we age, stem cells will actually fall into deeper and deeper levels of this dormancy until eventually in old age, they actually so deeply asleep, they can resist the activation cues coming from the brain. New evidence sort of tells us that it's defects in this quiescence or even in that adult neurogenic process that might sit upstream of some of the more classically recognized indicators of neurodegenerative disease like Alzheimer's and Parkinson's disease. So we're starting to understand that the regulatory mechanisms of quiescence are really clinically important, but unfortunately it's very tricky to study and we don't understand a lot of the mechanisms regulating quiescence, particularly those driving that increase in depth with age. Now, as you can imagine, studying this in human tissue is incredibly difficult because quite hard to find well-preserved human neural tissue. And so instead, our lab uses the healthy mouse hippocampus as a model system. It's actually a really well proven out model system for studying neurogenesis in the brain. And it's great because mice and humans have a lovely genetic overlap. The mouse age span is really compressed, so we can study aging in a realistic manner. That's also really handy because mouse and human neural stem cells share the same pattern of behavior. They're really highly active in early life. they enter a shallow state of quiescence, and that quiescence gradually deepens into advanced age. So it's a really nice system to actually answer some of these fine-tuned regulatory questions in. Now, a lot of what we currently know about quiescence has largely come from mouse genetic studies or single-cell RNA sequencing studies. And what's been really interesting from these studies is that it hints at cell-to-cell interactions being really important for regulating quiescence. What we see is that when ligands such as BMP4s or TGF-betas bind to receptors on stem cells, we actually see them exiting the cell cycle and entering a quiescent state. And conversely, when we see members of the Wnt families or other mitogens like EGFs binding to receptors, we actually see them exit quiescence and begin to replicate again. Unfortunately, what neither type of those studies can do is actually close the loop on that regulatory mechanism and identify the source cell sending that signal. So we're not really sure who is regulating stem cell behavior. Actually, very few cell-to-cell interactions in the hippocampus are well established. And so our lab hopes to get at this question now that we have the technological capacity, because we have single-cell spatial transcriptomic technologies, to identify the kind of source of that signaling driving stem cell behavioral change. Now, we use the Xenium Prime platform to answer these questions, and we're really focused on 3 key questions. We'd like to understand which genes and pathways in the niche change with age. We'd like to understand which hippocampal cells are interacting and how those interaction networks change with age. And lastly, we're interested in establishing which cell-to-cell interactions directly affect stem cell quiescence, and if it's changes in those interactions over time that regulate quiescence depth change. Down the bottom is what a hippocampus looks like when you profile it with Xenium. So you have a young mouse hippocampus and an aged hippocampus. And I just thought I'd point out the niche for everyone who's maybe not familiar with this region of the brain. So in the hippocampus, you have a dentate gyrus, which is this kind of duck-looking object here. And the red cells are that kind of thick granule cell layer that protect the DG. Looking at the stem cell niche, it's this very thin heterogeneous layer that sits between the granule cells and the hilus, which is the black middle. And you can see just visually that between a young mouse at 1 month and an aged mouse at 18 months, that stem cell niche looks very different. So it's a really nice technology to use to answer these questions. Now, we knew in trying to understand this very fine-tuned mechanism, we'd need to establish a fair depth of data to get around these questions. And while we would love to profile from birth to death, any single-cell spatial technology can be quite costly. And so we needed to pinpoint the actual points in time that would be worth profiling. Now, our lab and others have actually shown that it's between 1 and 6 months of age that our stem cells undergo the biggest shift in behavior. At 1 month of age in a mouse brain, we have a real abundance of stem cells. They're highly proliferative, and the ones that are in a quiescent state are in those shallow states of quiescence. But by 6 months, this has really changed. So there are far less stem cells. We're already seeing that reduction in stem cell number. They're far less proliferative than they were at 1 month of age. And the ones that are in quiescence are beginning to acquire those deeper quiescent states. We also knew that we would need to optimize lots of different aspects of the Xenium experiment, particularly around tissue preparation, because it is such a small region of the brain. And so we decided to include a reference point for optimization of an 18-month-old mouse hippocampus. Now, stem cells in the 18-month-old mouse are incredibly rare. There's very, very few of them. But the ones that are there are exhibiting the deepest states of quiescence. Now, we can't really get single cell type population data on stem cells when they are that rare. We'd need like a literal boatload of Xenium slides. But they are a really nice reference and really great optimization for our experiment. But we chose to focus largely on building technical replicates of 1 and 6 month-old mice. So this is what an experiment looks like in our lab. As I said, we have our pilot cohort, which profiles our 1 and 18-month-old mice, and our experimental cohort, which profiles our 1 and 6-month-old mice. And it's here that we actually ran 4 Xenium slides on these same biological replicates, just getting lots of technical replicates stacked there. To maximize capture on the Xenium slide, we actually microdissect the hippocampi out of the coronal sections of our mouse brains. And then we tile our hippocampi into FFPE blocks so that we have 1 hippocampus per replicate for each cohort in a single block. And it's actually a slice of that block that we analyze. Once those transcripts have been read, I take the tissue off of the xenium and I immunostain it for markers of stem cells and immature neurons. Now, this has actually been one of the biggest benefits of xenium for our lab. In whole transcriptome data, separating neural stem cells cleanly from astrocytes can be really difficult because they have a very similar transcriptome. When you're only looking at 5,000 genes, that problem gets compounded, so it's really difficult. We're able to get around that in xenium because it is compatible with this immunostaining, and stem cells have a really unique morphology and live in a really distinct region of the hippocampus. So we can take these confocal images and overlay it onto our cell data and manually annotate our stem cells so we know we have a clean ground truth neural stem cell population to actually analyze. We also knew going into xenium that we really wanted to focus on those cell-to-cell interactions. And while the prime panel definitely has signaling genes like receptors and ligands, it's pan tissue, so it's not hippocampal focused, and it's not really enriched for those signaling genes. And so we took advantage of the fact that you can run a custom gene panel alongside prime, and we designed a 100 custom panel that really focuses on those ligand and receptor genes that are enriched in the hippocampus. To generate this data set, I analyzed 4 publicly available data sets of different types of sequencing, looking for genes that were enriched in the hippocampus with age, or enriched in the neurogenic lineage. And then we whittled those down to the top 100 that we thought would be most likely to regulate stem cell quiescence. We actually ran the custom panel alongside our experimental cohort, so our 1 and 6 month old mice, which we just finished in February. And this is what that looked like. So I've taken out all of the base panel reads here. So each little dot represents a single custom probe. And you can see that they're really specific for the dentate gyrus, that duck-shaped object, compared to the background. And when you zoom into a little region, you can see that those probes are really nicely distributed across the different cell types. And that's really important because that means we'll be able to capture not just cells receiving signals, but also a plethora of cells sending signals. So really surprised at how well the custom panel worked. We actually designed it in collaboration with James Fraser, who's the technical rep for 10X for us. And the process was really easy and it's worked really well. So we finished that process in March of this year, profiling our last experimental run. And we've merged all of our data sets together. So if we spread out our dentate gyri on a table, just in 2D, it would look very much like this. You can see our pilot run at the top, which is our 1 and 18-month-old mice. We ended up with a fair amount of tissue damage here, so a lot of things that we optimized from this run to get to our experimental runs, where we ended up with a really lovely sectioning. So of our 18 mice in our experimental run 1, we had 16 in frame on the slide, and all 18 on the slides for run 2. So really lovely capture here. When we look at our data in UMAP space, you can see that we have this really classic xenium UMAP where we have slight interconnectedness, but in unsupervised clustering, we actually have really strong and distinct clusters that are really easy to annotate using canonical marker genes. Now, there are definitely challenges working in the hippocampus, but one of the really great things is that it's an incredibly well-defined region of the brain. So we know what cells should be exactly where. And with Xenium, you're actually able to pass this annotation back into Xenium Explorer and overlay your cell annotations onto the tissue. So we can check visually that all of our cell types are where they should be. And this is really important to us because profiling those cell-to-cell interactions, we need to have very faithful cell annotations. So it took a while to get this optimized, but we have really lovely, very well annotated, at least I think, data to move into answering those key questions. And we started answering these with key question 1, which genes and pathways in the niche environment change with age. And to do this, we take a leaf out of bulk RNA-seq and we use a pseudo-bulk approach. To do this, I start by manually demarcating the stem cell niche from each of my samples using Xenium Explorer, and then we aggregate the expression of those on a per sample level. So we basically take forward all of the purple cells in each section for analysis. And we do our pseudo-bulk analysis using Limma and the Voom workflow. We do use a duplicate correlation just to make sure we're not adding any pseudo-replication issues because of our technical replicates. And when we fit a pairwise comparison, which just basically looks at the genes that change between 1 and 6 months, 1 and 18, and 6 and 18, we see that we have a lovely split of genes that are upregulated with age and genes that are downregulated with age. And that's kind of despite the fact that we lost a lot of sections in that 1 and 18-month-old comparison. And we still see genes that are differentially expressed between 6 and 18 months, which is really exciting considering that power loss. So very strong transcriptional changes occurring in this region at that time. But those pairwise comparisons alone aren't particularly informative or even exciting. But having those 3 age groups means that we can fit a different type of model called a linear trend analysis. And what this does is it looks across all 3 age groups, our 1 month, our 6 month, and our 18-month-old mice, and asks what genes change across all 3 time points. Here's just a quick summary of the top 50 of those for up and down regulated, and you can see that we have a set that become progressively enriched with age or progressively down regulated. And what we can do here is actually take these differentially expressed genes and run them through a machine learning approach called k-means clustering. K-means clustering works a little bit differently to differential expression analysis. What it does is it looks at the change in shape of that gene over time, and then it clusters genes into specific programs based on that shape change. What we found is that in the niche between these 3 age groups, we have 5 temporally distinct transcriptional programs that change with age. We have 1 gene set that becomes progressively downregulated, 1 that becomes progressively upregulated with age, and then my 3 favorite, the gene set that becomes enriched between 6 and 18 months, 1 that decreases early, so between 1 and 6 months of age, and 1 that increases early between those same 2 time points. And it's really these last 3 that we're particularly interested in kind of drilling down into for those more fine-tuned regulatory mechanisms. We started by looking at which processes relate to these gene regulatory programs, and we started with early decrease in expression since we really should be seeing genes that relate to what we see biologically. Decreases in stem cell proliferation, decreases in neuroblasts. And what we saw when we ran gene ontology is that kind of mirror. So we see that the genes in this program regulate neurogenesis, cell fate commitment, neuroblast proliferation, precursor cell proliferation. And so this was a really nice control for the k-means clustering and reassured me that what we were pulling out of this analysis was biologically true. We then looked at the early increase in expression gene set, so those genes that spike between 1 and 6 months of age. And here we see early indications that cell-to-cell signaling becomes important between these age groups. So a lot of genes in this set regulate neuronal communication through synaptic signaling, but also other cell-to-cell interactions, such as through G-protein coupled receptor signaling or secretion by tissue. We also had a look at those genes that spike between 6 and 18 months and what they're regulating. And here we have kind of a much more complicated set of processes that I'm still trying to tease out a little bit. We get a lot of regulation of pH, things passing across the membrane, and lots of indications that it's neuronal heavy. This is where we've kind of paused key question 1 for the moment, while we wait on the integration of some single nuclear RNA ATAC data that we have that's time point matched to see whether these transcriptional programs correlate with chromatin remodeling programs. In the meantime, though, we've moved on to key question 2, which really looks at which hippocampal cells are interacting and how those networks change with age. Now, to do this type of analysis, we use a package called SPARTalk, which looks at the expression of ligands in cells sending a signal, as well as the expression of receptors and downstream target genes in cells receiving signals to call an interaction in a spatially restricted manner. When we look at the hippocampus, we see that it's an incredibly loud signaling environment. There's a lot of interactions passing between cells. On average, we see about 13,000 unique interactions in each hippocampus, which is far too many for us to look at in detail. And so instead, we decided we'd look at the interactions that are only seen in our 1-month-old tissues or only seen in our 6-month-old tissues. And we still get a lot of interactions. Just from the core diagram, I hope you can appreciate that at 6 months of age, it's a much more complicated, loud signaling environment than we're seeing at 1 month of age. It's actually about double the amount of interactions that are occurring at 6 months only than at 1 month. But again, this was really too many for us to look at. And so we decided to zoom in just to our population of interest, our stem cells, and see what their age-locked interactions look like. So we focused just on the 1-month-old, interactions that are occurring only in our 1-month-old tissues being sent or received by a stem cell. And we see that interactions that only occur here are very, very proliferative. Lots of proliferative chat directed at 1-month-old stem cells. And we see the first indication of gentle quiescent signaling occurring here as well. When we look at the age-locked interactions in our 6-month-olds, we see that the environment is much more balanced. They're still receiving unique proliferative signaling, but we now see an increase in pro-quiescent signals. And these signals are generally the ones that we associate more commonly with quiescence, like JAG through Notch, ApoE, and APP signaling. What's really interesting is that, again, this list is about double the length of the interactions that only occur at 1 month. And so there are a lot of interactions arising after this time point that are only seen at 6 months. I was really interested to see whether this was a change in transcription behavior of a static neighborhood, or whether this change in signaling behavior represents a change in the neighborhood composition around a stem cell. And so we went back to our base object and did some niche analysis. So in the middle of these pie charts, if you can imagine a stem cell, a quiescent stem cell, its neighbors at 1 month are predominantly made up of granule cells, which makes sense because they are the dominant population in the niche. But it also has a significant neighborhood of astrocytes, oligodendrocytes, and some neurons. At 6 months, that neighborhood is very different. So again, we've got our stem cell in the middle, but it's predominantly granule cells neighboring it. We've seen a reduction in astrocytes and neuroblasts and almost all other cell types. What's really interesting about this is that we found that there's almost perfect correlation between these changes in neighborhood composition and cells sending signals to our quiescent stem cells. We see an increase in oligodendrocyte signaling at 6 months, an increase in granule cell signaling, and a decrease in all of these blue cell types signaling to our stem cells. At the moment, we're working on understanding these interactions that are shared between these two age groups, but that are upregulated with age. And that will help us really shortlist candidate interactions that we think might be regulating stem cell quiescence for our future direction, which is to move into key question 3, looking at those signals that are directly affecting stem cell quiescence depth. What's really cool about SparTalk is that you can actually pull out from each interaction the downstream transcription factors that are affected by the interaction. And through Cell Oracle, this program that's summarized down the bottom, we can actually do an in silico perturbation screen, which means we can either knock out or overexpress that transcription factor computationally to see whether knocking it out or overexpressing it drives a stem cell into deeper quiescence or towards activation. And that will really help us shortlist interactions for in vitro validation. We'd also like to integrate again that single nuclear RNA ATAC data here to look at whether cell-to-cell interactions are regulating quiescence depth change via chromatin remodeling. So lots of things going on in this kind of part of the project, and we're hoping to present some more data to you at the end of the year. If anyone's at ASSCR, please come and say hi. But for now, I just wanted to thank everyone who's been involved in the project very much, my supervisors and lab group, as well as the scientific services team at QIMR, and of course, Millennium and 10X for giving me the time to speak today. Thank you so much.
Thank you very much, Stephanie. Are there any questions for Steph and Brett? You can unmute yourself and ask the questions, or you can also post them in the Q&A at the bottom of the chat. I'll ask a quick one. I have a comment and a small question. It's really amazing, like how you progressively improved to some tissue damage to mounting 18 hippocampal sections. That was really cool. I have a question around like SparTalk. It's really amazing that with the data that you can actually understand cell-cell interactions, but with the neural cells being all sorts of shapes and sizes and lengths, I wonder if that was a bit challenging to understand really which cells are interacting. Did it pose any difficulties?
So unfortunately, with our xenium data, we can't actually track the processes of cells. The dentate gyrus is really, really thick and very dense. So the processes of our stem cells get lost in the granule cell layer, and effectively, kind of the transcriptome of that gets incorporated into the granule cells. So we always need to be aware of that when we're analyzing the data. So really what we focus on is just the expression in the nucleus or very close to the nucleus, since we use a 4-micron expansion model for segmentation. So if there are, I guess, projection dependent interactions, we aren't able to pick them up. We're only picking up what's happening with the cell body.
Yeah. I have a question in the Q&A.
Yeah.
So it's from an anonymous attendee saying, sorry if I missed this. Why you lost some of your samples in the first trial? Do you have any tips of putting samples on sections? We're also working on small tissues.
Cool. My biggest tip is just to work really closely with your histology or whoever's doing the sectioning for you. So in our pilot cohort, we were sectioning our hippocampi to 500 microns first and then dissecting, and that's quite thin. When you are working with an FFPE block, you need to face the block and then take a few sections off before you can cut anything for xenium. Unfortunately, what happened to us is that we actually ran a perfect experiment on our 1 and 18-month-olds, but the machine had an error. And so we lost all of our data and had to run again. And it was in running again that we had to cut through usable sections because their RNA quality would have been a bit degraded having sat in the fridge. And so largely it was sectioning issues. So just make sure you have as thick as possible the tissue to go into a block to start with, I think is really the best tip. And work super closely with them. Our histology team was incredible helping us here. Really good.
Great. Thank you. Yeah, if you have any questions, yeah, please reach out to Brett and Steph. And yeah, thanks for attending today. It was really great. The talk was amazing, Steph. And thanks, Brett, for presenting Atera. Obviously, we all are really excited about Atera. Yeah, thank you, everyone.
Thank you.
Thank you.
Thanks, Kartik.